That I'm probably one of the few things standing between you and spring break, so I'll try to keep things short and sweet today. But we do still have some material to get through, and I promise it'll be painless and maybe even a little bit of fun. So I like talking about population regulation today because we're really going to be integrating a lot of what we've been talking about over the last several weeks, where we're going to think about what regulates the population size and how do we think about what controls from a top-down perspective versus a bottom-up perspective. In a nutshell, that's really what we're talking about today. And so it really does integrate a lot of different aspects of what we've been talking about over the last several days. First, the sun is the ultimate source of energy for most ecosystems. I highlighted this principle today because if we're thinking about what regulates populations from the bottom up, we're thinking about resources. So where are those resources? How much are there? And what does that mean for the organisms that rely on those resources? We're also still thinking about this equation, our fundamental equation about the population size at any given time. It's related to births and deaths and immigration. And then today we're talking about interactions among organisms. We're starting to layer this on top of each other and thinking about things and how they're organized into these webs of interactions. So in any given time, in any given ecosystem, these interactions are all happening at once, and so we need to think about them simultaneously, right? So how do the amount of plants affect herbivores? How does the amount of herbivores impact predators? How do predators impact things from the top down? Organizing those things requires thinking about these webs. Okay. So the specific topics we'll cover are, as I mentioned, this bottom-up model and top-down model, and how these go into this idea of trophic cascades. This is a pretty fundamental topic in ecology and something you may have heard about before, but we're going to be talking about some examples of each of these today and discussing how understanding these types of interactions help us to predict the population size at different levels within a ecosystem. So this bottom-up model, we'll start with that. This is the prediction or observation that in many cases the ability of a food or a resource or prey to regulate the next level up, so to speak, of a predator or the herbivore, that is thinking from the bottom up. So how do we track the amount of resources in an area to the herbivore or predator that depends on those resources? That's thinking from the bottom up. We can also, on the flip side, think about this from the top down as well. So how do we use the number of predators, for example, to predict the way that natural enemies, so this would include predators but also parasites that we talked about last time, how do they control prey abundances? And then how do these interact together to develop what we call trophic cascades? Okay. Any questions so far about those topics? It's going to be in broad strokes. All right. So to back up for a second, what we're talking about today might look something like this in a very general sense, where we're going to be tracking different trophic levels. When I say trophic levels, trophic comes from a Greek root word meaning to nourish, and so it's basically where's the food coming from at a given level within an ecosystem. So we can then categorize these trophic levels based on essentially who eats what and where they get their energy from. So here's our original second principle of ecology. Sunlight is 1 million joules in this diagram. So all that energy is coming from the sun. We then go to the next trophic level, which is primary producers. These would include anything that's autotrophic, mostly plants in this case. And we can see that they contain some energy which they're getting from the sun, right? So through photosynthesis, they are gathering that energy, turning it into biomass. That biomass also contains energy. And that's all located in the trophic level that we call primary producers as a whole. If we go one step further from that, we have our primary consumers. These are also known as herbivores or detritivores. So they are eating plant material, either dead or alive, and gaining energy from consuming that biomass or necromass, as it were. Now, these primary consumers also contain energy, of course. They are chemical machines that run on that energy, but they contain it as well. And so they're supporting our secondary consumers, which would be predators. They're eating things like insects, which are herbivores or primary consumers. And then we can also go up from there, and we have tertiary consumers, things that would eat with secondary consumers. So all told, we have 5 or 6 levels that we need to be concerned about when we're looking at these effects from the bottom up, so a resource-based perspective versus the top down, which would be a predator-controlled perspective. Are there any questions about these categories of trophic levels? So in a couple weeks in the lab, we'll get to go out and document some of these patterns at Glacier Creek and look at these different trophic levels and what that means for the food web out there. So just to reiterate this idea of bottom-up. Bottom-up means that in this scenario, the base of the food web, so the resources, including sunlight or nutrients, and then the primary producers that rely on that light and nutrients, is what controls the overall population of the next level up. So we can follow it from the base of the food web up to the top of the food web, and ultimately it's the resources that are controlling the populations at each step of the way. So that would be a bottom-up perspective. That means that from the top down, this means that we're thinking about this from a predator down perspective, and that the number and type of predators within an ecosystem is what ultimately controls the trophic levels below. So we have these two kind of competing viewpoints, and they're often happening simultaneously together, and so we have to consider both of them in any ecosystem that we're going to explore. Right? So how do these bottom-up effects and top-down effects play out in nature? And what does that mean ultimately for the abundance of populations within each trophic level in an ecosystem? So let's go through some examples that show experimentally as well as observationally what happens in nature if prey or food resources are what's regulating population size of herbivore and predator abundances. So we'll first take this example of water hyacinth and weevils. This is an example that's outlined in your textbook, and we're going to go into detail here today as well. So this water hyacinth is a aquatic emergent plant, and then there were 2 species of weevils that were studied here, and basically the setup of this experiment was to examine population size and other population variables that we've been thinking about in relation to the nutrient content of this primary producer, in this case water hyacinth. So in this experiment, what they did was they grew up the water hyacinth, our primary producer in this example, under different levels of fertilizer application. So in each of these little tanks or mesocosms that they had with water hyacinth, they fertilized them with either low, medium, or high rates of fertilizer application. And at each higher level of fertilizer, they saw this increase in the amount of nitrogen that was contained in the tissues of this water hyacinth leaf. Okay, so more or less, you add some nitrogen to a tank where there's plants growing, they take up more of that nitrogen into their biomass, and then it's incorporated into that leaf. All right, so that's what both of these graphs are showing in general, that across these treatments, across these different tanks, if you add fertilizer, the plant is going to be more nutrient rich. That make sense so far? So they took this one step further and they said, okay, if we add some fertilizer to these tanks, and then that's incorporated into our primary producers, what does that mean for the populations of an important herbivore for this plant? And overall what they found is that when they grouped together the hyacinths in low, medium, and high treatments of fertilizer, they found a higher percentage of reproductive females and overall higher abundance of female weevils especially in the high nutrient treatment compared to the low nutrient treatment. Okay, so there were some nuances here to think about, some differences among the two different species, but for the most part, what they're seeing here is that if you add nutrients and that gets incorporated into primary producers, then that leads to an increase in the abundance of an herbivore. So this is pretty strong evidence for a bottom-up effect of increasing nutrients, regulating the population of an herbivore that depends on the nutrient content of these plants. All right? Does that make sense? Okay. So let's go back to parasitism for a second. And just as a reminder, we've already kind of talked about this bottom-up effect and how it can influence the populations of important pathogens, right? So one of the ways that we talked about this was with the malformed frog example, where on Tuesday I was talking about this in the context of global change, but it's important to recognize that this global change, where we're adding nutrients to the environment through fertilizer runoff, is inducing a bottom-up effect, right? So we're adding nutrients. In this case, the experiment was to add nutrients. This is happening across the landscape in ponds all over the world. If we add nutrients, then in this example, that boosted the populations and survival of this parasitic trematode that was infecting these frogs. And this suggests that eutrophication, or increased nutrients from the bottom up, can influence pathogen abundance within ponds in the US at least. We also saw this when we looked at plant pathogens, and when we compared ambient nitrogen to when fertilizer was added, the pathogen load on these plants within these plots tended to be higher when we added nutrients. Again, suggesting that pathogens can be controlled, at least in part, through a bottom-up pathway, where if we add a resource that then influences something about the plant that then is increasing the abundance or prevalence of a pathogen, in this case, a plant pathogen. Questions about those two examples? Okay. The third way we already talked about bottom-up effects is with the ticks and the time lag that we saw between resources in the form of acorns from oak trees, the host for the ticks, which was the populations of these ground squirrels, and then the tick populations themselves. So if we follow these trajectories through time, the time series suggests that from the bottom up, the abundance of ticks and their prevalence is highly dependent on their host abundance, and the host abundance is affected by the number of resources in terms of their food. So this herbivore is being affected from the bottom up, and that in turn is also affecting the abundance of this parasite. Okay. So those are three examples we already talked about. I'm just telling you now that there are also important examples of how bottom-up effects can happen and their implications. But I'm going to tell you about this example today, and this is an example that's close to my heart because it's related to my dissertation research. This is a picture of one of the streams where I did my PhD work. It's at Coweta Hydrologic Lab in Western North Carolina. And what we set out to do in these experiments was to test how bottom-up control can affect many aspects of this ecosystem. And today I'm going to tell you about an example where we looked at how adding nutrients to an entire stream can change the abundance of primary consumers within that stream, but not predatory consumers within the stream. Okay. So the basic setup was we added nutrients to an entire stream reach, or a section of a stream. And we did that by capturing water from upstream and then putting it through these pipes. And then we injected some fertilizer into an irrigation tube that went down the length of an entire stream. And that boosted concentrations of nutrients within the stream water, but not really anything else. So we manipulated basically one aspect of these streams where we were bumping up the amount of nutrients that are available within the ecosystem, but it was only available in the water. So there was some pathway through which it was being entered into the ecosystem that had an effect on the abundance of other consumers within the stream. The way this pathway turned out to work was when we added nutrients that stimulated the growth of aquatic fungi, which are pictured here, they're a little hard to see. These aquatic fungi grow on dead organic matter like leaves, and they tend to also take water column nutrients and then store them on the leaf surface, making them more nutritious for detritivores that live in the stream. So through this bottom-up pathway, we saw that by adding nitrogen and phosphorus directly to the stream water, that increased the decomposer ability to sequester those nutrients on dead organic matter in the stream, that increased the nutrient content of that leaf litter, similar to what we saw with the water hyacinth. And that in turn, we think, led to increased populations of primary consumers, these aquatic detritivores that look something like this. One of the more important detritivores that tended to respond most to adding nutrients from this bottom-up pathway was this caddisfly that builds a case. It's called Pycnopsyche, and they're quite large. They can be a couple inches long, and eventually, once they get to be large enough, they start building a case out of stones. And those cases built out of stones are meant to, or do help them avoid getting eaten. And that has important implications for the rest of the food web, as we'll see here in a second. So what we were able to document, or in a previous study that I wasn't involved with, but was in the same system, what they found was that if you add nutrients to a stream, you stimulate the production of this important detritivore, but because of their size and their ability to make a case, they don't end up getting eaten by predators, and so we don't see a correlated change to the production of predators within the stream. All right, so with this graph, what I'm showing you is the primary consumer production, so how much or what's the abundance of our detritivores in the stream. You can see that when we added nutrients, we bumped that out way up here to 30 grams per meter squared per year, which is, it doesn't sound like a lot, but it is for a bug. So it's a lot of bug that's being produced by adding nutrients to the stream. Now we'd expect, if we're following this bottom-up pathway through to the predators of these primary consumers, that if we increase their production or their abundance, that the predators should also increase along this line. Okay, so this solid gray line here shows the relationship between all of these points, which are in non-nutrient or ambient streams that don't have nutrients added. Okay, so there's a pretty strong relationship between the amount of prey that are available in these streams and the amount of predators that are produced within these streams. So their abundance are almost matching up pretty strongly. But we see that when we add nutrients experimentally, the detritivores, particularly because all of that resource is being tracked into this Pycnopsyche that is resistant to predation, what that means is that we're not having the same relationship happening in our nutrient-rich streams as we do in the control or ambient streams. Another word for this is called trophic dead end, where all this bottom-up energy that is being stimulated by adding nutrients from that pathway is not going up to the entire or top of the food web, right? So this predation-resistant taxa is more or less a dead end in the food web, and this increase in resources is not playing out throughout the entire ecosystem. Questions about that experiment? Okay. So let's turn our attention to the top-down model and how that differs from the bottom-up. Not just in sort of the topographical, thinking about it in a diagram sort of way, but also what does that mean from an ecosystem standpoint. So this is a top-down example that's very famous from an experimental lake system. This is a lake that is in Canada. It's in the experimental lakes area in central Canada, I believe. And this is an area where they were able to conduct entire ecosystem experiments, and in so doing learn a lot about the effects of basically removing predators from one ecosystem and keeping them in another one. So in this experiment, the basic idea was they took out a predator, in this case a largemouth bass, and they totally removed the entire population of largemouth bass from one side of the lake, and then they put up this little barrier in the middle in this kind of narrow part of the lake, and then the other side they still had bass. So based on this picture, are there any guesses about which side had largemouth bass and which one didn't?
Largemouth bass on the left.
Got your guess? Okay. So if you guessed the one on the right, then you were correct. Sorry, that's the one where they still had the bass. Let me back up for a second. If you guessed the one on the left, then you are correct. That's the one where they removed the bass. And you can see that from this aerial photography because what's happening here is essentially a trophic cascade. But the trophic cascade is being regulated by the presence or absence of this predator at the top of the food web compared to at the bottom. All right, so the way that this works is what we see here is this fish, which we call a zooplanktivorous fish because it eats zooplankton, like our friendly little Daphne over here. So it's eating the zooplankton, and those zooplankton would be eating the algae that are growing in the lake, but because their population is being regulated from the top down by this planktivorous fish, that allows for algae to grow unchecked. And so there's this indirect effect of the planktivorous fish on the amount of algae within the lake that is happening because there aren't any predators controlling the population of this planktivorous fish. Right? So if we take this side of the lake in this experiment, where the bass were still there, the bass are controlling the population of the smaller fish that eats plankton. So that means that the plankton are allowed to have higher abundances, so their populations are not kept in check by the planktivorous fish. And then because there's enough zooplankton around, they can consume all the algae. So this is our main herbivore in this ecosystem. They are consuming algae and keeping its population size in check. So in this case, it's another indirect top-down control example because the bass itself is indirectly controlling the population of algae through its direct control on the population of planktivorous fish. All right, so this is a very classic example of top-down control causing these indirect effects that then cascade down to be observable from the air in terms of the algae that's growing on within a lake. There are lots of examples of this, and we'll talk about a few more of them. This is one that maybe you've heard about. It's been in the news. It's related to the top-down control of urchins by otters and how prey switching from these orca is reducing the number of otters, and those otters are then not controlling populations of urchins. These urchins will mow down the numbers of kelp, and it's having a strong impact on our kelp forests in coastal marine systems. So another example of an indirect effect of a top-down control that is controlling our primary producers through its effects directly on the predator of that herbivore. All right. Any questions about those two examples before we move on? Okay. So what I'm going to show you here takes into account what we've been talking about and kind of combines these ideas of bottom-up and top-down control. So if you remember, I said that these are both happening at the same time, likely in any ecosystem that we're thinking of. And so what this study does is summarize a number of experiments where they tested top-down effects versus bottom-up effects. And they compared the strength of these bottom-up versus top-down effects across 23 different studies. So in this top graph, what we're looking at is bottom-up effects by themselves. These are basically fertilization experiments where they went out and they added some resources to an ecosystem in the form of fertilizers, and then there is also a summary of top-down effects where there's been a predator removal experiment. Okay, so those are kind of the two different experiments that they're summarizing here. And on the y-axis in both of these, what they're looking at is, on the top one, the fertilizer effect. And the way that they do this is to compare the experimental treatment where they added fertilizers to the control treatment where they didn't add any fertilizers. If that number is greater than 1, or in this case greater than 0 because they log transformed it, if it's greater than that threshold, then that means there is a positive influence of the fertilizer. If it's less than that, then that means it's negative. Here, we're looking at the same thing just in terms of predators. So they're comparing the treatment where they had predators added versus a treatment where predators were excluded. And then they looked at the overall effect there as well. Again, there's a threshold where if it's above 0, it's going to be a positive effect. If it's below 0, it's going to be a negative effect. So what they found is that if we look at these 23 different studies and across all of them, and we summarize them, on average, the effects of removing predators tends to have a stronger effect on herbivores than fertilizer effects do from the bottom up. Okay? And then the other piece of this is that adding predators decreases herbivores and increasing plants, but adding fertilizer does not significantly increase herbivores on average. Okay? So again, we're summarizing many different studies here. They're looking at both of these pathways at the same time. And the upshot of this is basically that there's stronger top-down effects that can be detected in these experiments compared to the bottom-up effects. Questions about this summary? So we already kind of talked about this trophic cascade idea, but I'd like to dig into it a bit more in a general sense and not apply it to an example where in the trophic cascade, basically this means that you have top-down effects that quote-unquote cascade to lower trophic levels. And so the top-down effect that we've been talking about so far is that there's this direct interaction between a consumer and its resource, where if we're looking at that from the top-down perspective, we expect the consumer to reduce the amount or abundance of their prey in some way, whether it's a plant or an animal. We expect the top-down effect to be an increase in the consumer leads to a decrease in the basal resource, whether that's prey or a plant. When we add on a third level, to this interaction, then there's an indirect effect that's introduced that we need to be aware of that is also known as this trophic cascade. So another way of saying trophic cascade is that we're documenting an indirect control of a lower trophic level through this top-down effect, right? So it's a similar direct interaction here where the predator is going to increase and then decrease the consumer. So this interaction is similar to where we're just looking at this one, but by controlling the consumer population, that means that indirectly they are allowing the primary producers or the basal resources to flourish. Okay, so that's where that indirect effect comes in. And so I hope that at this point you can explain the differences between top-down effects with and without a secondary consumer or a top predator. So in other words, can you explain what's going to happen if I tell you that in one ecosystem there are 3 levels to be concerned about: a primary producer, an herbivore, and a predator, versus another ecosystem where we have primary producer, herbivore, predator, and then secondary consumer or a top predator. Okay, so that's basically the difference between the 2 lakes that we discussed earlier. What happens to each trophic level? Being able to draw an arrow in terms of how those are going to affect each other. So if this goes up, this goes down, and this goes up, sort of alternating more or less. If this goes down, this goes up, this goes down, this goes up. Is there any part of this that's kind of unclear still, or you feel pretty comfortable with it? Comfortable? Okay. Cool. So you may be wondering, well, okay, if this is happening all the time, that's great and all, but what is sort of the implication of this top-down control and its strength for regulating populations at multiple trophic levels within an ecosystem? Well, part of this that we need to be aware of is that these types of top-down interactions are becoming less common because of our outsized effect on many different ecosystems and the top predators that live within them. So this is known, or has become known, as trophic downgrading of Earth, where we're basically lopping off the top of our food webs through various changes that we've made in our ecosystems. One example is sea otter and kelp, which is pictured up here. So our interactions related to over consumption of, or an exploitation of seal populations has led to orca seeking out less preferred prey, in this case sea otters. And then those sea otters are no longer able to control urchin populations, and that leads to downgrading of our kelp forests. Another example, which we'll talk about in lab after break, is pisaster and mussels. Pisaster is a sea star that lives in the rocky intertidal zone. It's also known as a keystone species. We'll talk about that concept in a couple weeks. The idea here, though, is that it is susceptible to a natural enemy that's a pathogen, sea star wasting disease, and because of this disease and its prevalence, it is becoming less abundant in our rocky intertidal zone. And as we'll see in a couple weeks, that has really strong implications for its control of mussel populations in the rocky intertidal. We talked about the bass, zooplankton, phytoplankton example quite a bit. That's pictured here. This one is certainly affected by humans, especially with sport fish and sport fisheries related to bass. In this case, the interesting application here is that bass are sometimes introduced where they perhaps weren't before. Stocking those fish has implications for the food web of the lakes where they are introduced. What does that mean for the overall food web and functioning of these ecosystems? And then sharks and large reef fish, similar example here, indirect effects that are affecting our coral ecosystems. You can look at present and absent in terms of shark populations and their extirpation locally from reef ecosystems. And this is another cool example that I don't have listed here. This is in Great Plains streams, looking at bass and stone rollers and their ability to control algae populations in Great Plains rivers. It's not just in aquatic settings, so marine and freshwater. We can see trophic downgrading also occurring on land. And these examples, maybe you've heard about some of them. Here is the difference between having Arctic fox on the right and where those Arctic fox are absent, you get uncontrolled growth of grasses. This is presence and absence of jaguars in the jungle. This is presence and absence of wolves in Yellowstone. I'm guessing some of you have heard about this example perhaps before, where willow and riparian zones of streams in Yellowstone are highly dependent on the presence or absence of wolves and their ability to control elk populations that browse on these plants. And then we talked about wildebeest last time in the context of rinderpest and how that pathogen can control their populations. This is an interesting example where we have a natural enemy that is a pathogen controlling the populations of wildebeest, and that has implications for indirectly the primary producer populations in the savanna. Okay. Questions about trophic downgrading? So I suggest being able to take one of those examples and explain what's happening in any one of them. Okay? I guess I'll note again and emphasize that in each case, what we're talking about here is top-down control and the indirect alternation of the control of primary producers essentially in these ecosystems. All right, so to close out today, like I said, I was going to try to keep it short and sweet for you. We're going to talk about a really fascinating example that is combining both bottom-up control and top-down control in a single system, and how it can help us predict the prevalence of a zoonotic disease, in this case, Lyme disease. So we're going to take another look at Lyme disease as an example of how top-down control and bottom-up control are both at play in this system, and how understanding both of those together help us to predict the prevalence of this human disease. So this study came out in 2012, and basically the main message that they came up with was that restructuring populations of predators in the Northeast and where humans have basically built our cities and towns may have unintended consequences for human disease risk. And so that happens through, again, kind of a trophic downgrading that's happening in our forests, and what that means for host populations, and ultimately Lyme disease carrying hosts, these larval ticks. So the data that show this pattern across, in this case, 3 different states. We have New York, Virginia, 4 different states, I can count. New York, Virginia, Wisconsin, and Pennsylvania. Is what they looked at here were the abundance of deer, because part of this hypothesis is related to what's the abundance of deer, because the host is named after, let me back up for a second. So these bottom graphs are showing a potential host population, and they're looking at deer specifically because Lyme disease occurs within deer ticks. And they're so-called deer ticks because we often find them on deer. And so there's this hypothesis that perhaps increasing deer populations are related to the increased prevalence of Lyme disease in these different states. Is that better? Okay. You following me so far? So that's what they're trying to show here. And what you can probably tell is that there's not really a strong or noticeable relationship between the number of deer, their density and abundance, and the incidence of Lyme disease across these counties in these 3 different states. And the same is true in New York as well. So it's kind of just deer are here, and Lyme disease incidence is very variable across that range of deer population size, and there's not really a strong relationship between the two. Do y'all see that? Okay. But where they did see a strong relationship between Lyme disease incidence and a population abundance was with the population size of coyotes relative to number of foxes and the number of foxes that they found. Okay? So fox, we see this negative relationship between Lyme disease incidence and the number of foxes. So if you find more foxes in an area, you see lower Lyme disease incidence. Okay, so that makes sense, right? Because we think and we know that foxes are going to prey upon an important host for Lyme disease or Lyme disease carrying ticks, which are the white-footed mouse. So they eat a lot of rodents that would be potential hosts for the ticks that are hosting the Lyme disease. And so when you see more of them, you see less Lyme disease. Now there's this other fascinating pattern here where we look at the number of coyotes per fox. So where there are more coyotes per fox, or just more coyotes, more or less, we see higher incidence of Lyme disease. So what's going on here? What they were able to work out using these studies and understanding the natural history of these different predators and the way they interact together is that coyotes are very territorial and will actively remove any foxes from their hunting territory. So where there are more coyotes, they will basically extirpate or remove those foxes, and so you see more coyotes per fox in these areas. By reducing the number of foxes in these areas, that increased the population size of the host animal, in this case the white-footed mouse, which increased their viability as a host for the tick, which then increased the incidence of Lyme disease. All right? So that's when there's coyotes present. When coyotes are not present, then we see this interaction occur where the foxes are able to control the small mammal hosts and their populations, so you don't get this interaction between the parasite and the host. And then that control leads to lower Lyme disease incidence. So hopefully you can see by looking at the diagram in this way that we're basically comparing a 3-level food web versus a 4-level food web, similar to what we looked at before. In this case, though, the interaction is not one where the predator population is being controlled directly through a predatory interaction. What's interesting about this interaction is that it's a competitive interaction, right? So this is an interference between two predators, and due to the interference competition between the coyote and the fox, we see this indirect effect on the abundance of hosts that leads to increased Lyme disease incidence. All right. Did I do okay in the details of that one? Does it make sense?
Mm-hmm.
Pretty cool, right? I think it's cool. I mean, I don't know if it's cool from the standpoint of Lyme disease incidence, but I think it's a really clever application of food web, competition, and disease ecology all at once. And it really illustrates nicely, I hope, this idea of bottom-up effects versus top-down effects, and how that has a really strong implication on not just something like population size of, kelp or urchins, but also something that potentially affects us in our health. Okay. So I have some more study guide type of questions for you, which we can run through quickly, and then we'll get out of here. So be able to discuss your favorite example of a trophic cascade. I kind of mentioned that already. Think about what will happen if each trophic level is removed or manipulated in some way. Make sure you understand which tends to have a stronger response in terms of regulating populations. Is it top down or bottom up? At least based on that summary that I showed you. And then be able to discuss two differences between parasite host and predator prey interactions. So think about that table that I showed you, we walked through last time. What are the differences between a parasite and a predator in terms of things like their body size and their ability to regulate populations of their host or prey? And then think about this example, true or false, frog malformations are caused by increased contaminants in ponds. This is false. Right? It was a parasite. All right, well, I'm gonna leave things there unless there's other questions. And I hope you all have a great spring break.